Okay, so this is the next video. The question for number 3 is, we know Mary is a carrier of the cystic fibrosis allele, but does not have the disease. Summarize your findings for Mary's CFTR alleles. Describe the mutations that have occurred, which I already went over in the last video. Discuss how, if at all, they affect the protein, which I also went over in the last video. And explain how your genomic data fit with what Mary already knows, including which allele she must have inherited from each of her parents. Now, I'm not going to answer this for you because I feel like I already answered probably the whole thing, but the way to find the answer about which allele she must have gotten from which parent, it's pretty much at the very beginning of page 21, like the first page of chapter 2, the little in italics. It kind of tells a story about Mary and where she must have inherited her alleles. And you might be able to pinpoint on page 23 with figure 2.2 which mutation she must have and what that must mean for her. Okay, so what I'm going to do is I'm going to move on to number 4. It says repeat your analysis for each of Tom's 2 alleles, which in class we found out today means that we're supposed to pick up from detecting mutations. So there's a page 20 or page 30 where at the bottom it says detecting mutations, and there are these steps for detecting mutations that we're supposed to follow for Tom. Okay, so let's see. All right, repeat. And then it says, is he a carrier for cystic fibrosis? And summarize your findings and determine the probability that they will have a child with cystic fibrosis. Okay, so what we're going to do is we're going to go, I believe the instructions say, go to pairwise aligned codons. Pairwise aligned proteins, aligned DNA, pairwise aligned codons. Okay, we're going to put Tom's information in here, his original data in here, and we're going to put the wild type original data, not the translated, in there. So we'll do that, and then we'll come back. All right, I'll do that first, and then I won't look at anything else yet. Tom A. Oh, A. All right, and then I know I'm going to put wild. Okay, Tom's original A data. All right, I'm going to close out Mary, I guess. I can take wild. No, we don't need wild translated. Goodness. Okay. So, Tom's original code. Opening that up. Since we've got it, I'm going to highlight from above that position to below, so I know I've got everything. Tom's A. Paste, okay. And then we have the wild type, which we can open up our wild, not wild translated. Okay. Again, super important to do this whole thing with Notepad. Okay, go ahead and submit that. Give it a second. Okay. Alignment results for 4,440 residue sequence with Tom A. So Tom A has 4,440 and wild has 4,443. So if he has 440 and wild has 443, what can you assume already from the get-go? I hope I don't have to tell you what we have to assume from the get-go. Okay. All right, so we've got that information. Let's go ahead and write that down on your sheet of paper or whatever. Make a note of that, make a mental note. Okay, we're gonna go ahead and put in Tom's B allele. Tom's original code B. results were 443 for Tom B and 443 for Wild. Okay, so already you're good. Okay, so you know you're gonna line up just fine. Oh, didn't mean to do that. Did not mean to do that. Okay. All right. The next step for detecting mutations on page 30, because again, we're doing all this for Tom. All right. All right. It says, okay, what's the case? In this case, they line up base for base, it says, but that was for Mary. They would line up. It says, okay, so for Tom's B, they line up base to base. So there's 443 to 443. In the paragraph on the bottom of page 30, it says, though, if it is not true, you would see dashes representing the places where one sequence should be shifted over. Okay, or to visualize the results, copy both sequences, including their comment lines, and paste it into Color Align Conservation Tool. Okay, so Color Align Conservation Tool. Right here. Again, we're looking at Tom's A because that's our wonky one. That's the one, we're just looking at that temporarily right now because that's the one that did not align well with Wild. We'll compare it with Wild type. Oh, good. Already got it highlighted. Just copy again. Next line. Paste. Ooh, problem with alignment format. Dang. Forgot what to do here. Here, give me a second. I'll come back to this. I'm having a problem with alignment. I'm going to go back.